and I'm very happy to be here on this wonderful conference. My talk is, dealing with proteins, and proteins is a kind of biomolecules which are abundant. You find them everywhere. We humans, we have them from hair to toe, and you find them in all living species, including viruses, which are no living species, of course. So you find them also from viruses to humans. And why are they so important? Because they do nearly everything in living organisms. I have listed here some examples. So at the moment, they are busy with your breakfast and see to it that you get energy from your breakfast. But sometimes things go wrong and the proteins misfold. They don't find the right shape, they misfold. And this gives rise to so-called protein misfolding diseases. And they often involve short protein stretches, and then we call them peptides. So I will mainly use the word peptide, but a peptide is, in my context, is a small part of a protein. And one example is Alzheimer's disease, which is the most common neurodegenerative disease. It leads to the loss of neurons, as you can see here in the comparison between a normal brain and an Alzheimer's patient brain, which of course gives rise to loss of memory, and in the end, the patient dies. There's still no cure in spite of intensive research. And the disease is very demanding for the healthcare systems because of the long time that the people have the disease and because of the costs that are involved in caring for the patients. And this will increase tremendously because the number of patients is expected to triple until 2050 because we get older. And this disease is linked to the aggregation of a peptide, which is called the A-beta peptide. And this peptide, or as you can guess, the disease is of course very complicated, and there are lots of biomolecules around that interact with this A-beta peptide. And all these interactions influence how this A-beta peptide aggregates and then leads to the hallmark of Alzheimer's disease, which are the plaques in the brain of the patients. So that's why we decided to study interactions between the amyloid beta peptide, A-beta peptide, and other peptides. And one of the questions is, how is the aggregation of the A-beta peptide influenced by these other peptides? And therefore, of course, we want to study mixtures. One component is the A-beta peptide. The other component is the interacting peptide. And here you see an example, mixture of two peptides. But of course, in this AFM image, we don't know where is which peptide. And of course, that information is essential to find out whether one peptide influences the other peptide in its aggregation behavior. So we want to distinguish these two peptides, but we run into the problem. They are chemically very similar, so they are both peptides. And in addition, they have the same structure. So both of them like to form beta sheets. So there's no simple distinction from the infrared spectrum, and that's why we started to use isotopes. And the motivation is shown here. So here we have an A-beta spectrum with unlabeled peptide. The amide 1 vibration is seen here. And this is a spectrum of a 13C labeled peptide, which is clearly shifted. And this gives rise to the possibility to distinguish labeled and unlabeled peptide. So our approach is that we use two wave numbers at which we image the peptides. And I want to call them blue and red wave numbers. The blue wave number is characteristic for the unlabeled peptide, and the red wave number is characteristic for the labeled peptide. And when we do the imaging, then we expect a larger signal at the blue wave number for the unlabeled peptide than at the red wave number. And the opposite is true for the labeled peptide, where we expect a larger signal at the red wave number as compared to the blue wave number. Okay, so we did this, and this is an example now a control. This is unlabeled A-beta peptide. and you see the image of the phase at two different wave numbers, the blue and the red wave number. And you can see that the signals are stronger for the blue wave number than for the red wave number. And yeah, that indicates that the method works. But these phase images, they can be quite deceptive because the impression of the color depends very much on the choice of the background color. And that's why we wanted to have an unambiguous method to find out which of the signals is stronger. And what we did is we just subtracted the phase images at these two different wave numbers. But when we do this, of course, the images are not properly aligned, so we see parallel features, blue and red features. We have to align the images so that the features superimpose, which is shown here. And then we clearly see that we have mainly blue color here, as expected, for the unlabeled amyloid beta peptide. Yes, first comment here, please ignore this. This seems to be some dirt or impurity. It has a different shape, and the absorption is completely different from everything else. Then I said, okay, we have mainly blue color here in the image, but we have a region in this image which has some red color. So in a way, this seems to be a problematic region where the imaging probably didn't work so well. And I should say also that this sample was particularly difficult to align because we needed different shifts in different regions of the image to align the two images. So we then did another control, and we subtracted two repeats at the same wave number. And this difference image is shown here, and it's plotted in the same scale as this one. And you see that the features in this control difference image are much weaker than the features in the proper difference image taken at different wave numbers. Okay, fine. So it seems to work. And now I show you the first interesting sample. And this is a sample which is composed of labeled A-beta 40, which is a shorter version of the amyloid beta peptide, and unlabeled A-beta 42, which is the full-length peptide. And the preparation was such that A-beta 40 formed fibrils before mixing, and A-beta 42 formed protofibrils, that means smaller aggregates. And then we mixed them, and we calculated our difference spectrum, as you see here. And when we compare this with the mechanical phase, and we look, for example, in this region, then we see that the fibrils are red, which means that the fibrils consist of A-beta 40. And this is not only seen for these rather large fibrils, this is also seen for very tiny ones. Here you hardly see this in the mechanical. This little fibril, when we take a line scan across this little fibril, then we get these two line profiles, and you see that it has a stronger intensity at the red wave number than at the blue wave number, which means that it's labeled and which means that it's from A-beta 40. And there are more examples of the same fibrils, and they are all stronger in the red wave number than in the blue wave number, meaning that they are A-beta 40. But in contrast, these more round blobs here, the amorphous aggregates, they show stronger signal for the blue wave number than for the red wave number, which means that they are unlabeled and belong to A-beta 42. And also there we have several examples which all show the same. Okay, and now comes the real interesting sample, and that is where we mix the A-beta peptide with an inhibitory peptide called NCAM1-PRP. That is a peptide that is shown to interfere with the aggregation of the amyloid beta peptide and actually to relieve the cytotoxic effects that the amyloid beta peptide has on neuronal cells. We did a one-to-one mixture where this NCAM1-PRP peptide was unlabeled and the A-beta peptide was labeled. And I show you two different difference images where we clearly can see the locations of the amyloid beta peptide in red and the location of the unlabeled NCAM1 PRP peptide in blue. And what we see that the fibrils, when we analyze this image, also not only the different images, but also different line profiles, then we find that, where's the laser? Yeah, we find that the fibrils are always blue. That means that the fibrils are the NCAM1 PRP peptide, or they are gray, which means that the fibrils are mixed. And that could be because the NCAM1 PRP peptide coats fibers from the amyloid beta peptide. Because we don't see any fibers that are from the amyloid beta peptide, our conclusion is that this inhibitory peptide either coats or it dissolves existing amyloid beta fibrils. Okay, so that brings me to the conclusions. I hope to have convinced you that we can use 13C labeling to distinguish peptides which are otherwise not possible to distinguish from their chemical or their three-dimensional structure. And we started out with this image where the question was, where do we find which peptide? And now we can answer this question, and we can see that everywhere where we have red color, we have the amyloid beta peptide, and everywhere we have blue color, we have the NCAM1 PRP peptide. And the conclusion from this work was that this inhibitory peptide, the NCAM1 PRP peptide, coats or dissolves amyloid beta peptides, amyloid beta fibrils. Yes, and before finishing, so now something completely different. We also measured as a control just the purple membrane, and I want to show this to you because I would like to discuss the reason for the effect. We have recorded spectra, as shown here, of the phase and of the Nano-FTIR absorption, and the peak position is different for the different harmonics in the phase spectra and in the Nano-FTIR absorption spectra. And we have some explanation for this, but I would be happy to discuss this further with the experts in the field, whether you have observed something similar or not. And at least for the phase spectra, I think the effect is maybe a bit surprising. Okay, so this brings me to my last slide. From this work, the peak wave numbers, we found that the peak wave numbers differ for phase and Nano-FTIR absorption. This is not surprising, this was in the literature already, but they also differ for different harmonics of detection. And this work was a collaboration with Magnus Jonsson, who is going to talk a little bit later today, I think, from KTH in Stockholm, Astrid Gräslund regarding the peptides from Stockholm University, and Ramona Schlesinger from the Freie Universität Berlin. And all this would not have been possible without the generous funding of a private foundation, the Olle Enquist Stiftelse, which paid for the instrument and for 2 postdoc grants. And the work was also supported by Jan Fonden and Magnus Bergwald Stiftelse, and now also from the Swedish Research Council and from the European Union. And all the experiments were done by Suman Paul, who is now in the audience and is now employed by Neaspec or AutoCube. And Adela helped with the interpretation and the evaluation of the data. And I would also like to thank Adrian for his tremendous support and for never getting tired of answering all the questions, at least not showing of getting tired to answer all the questions that we posed to him. And also I would like to thank Sasha, who helped with the interpretation of the effect that the different harmonics show different peak wave numbers. And I thank you for your attention.